Alright, how's it going everyone? It's Chow here, and today we are going to talk a little bit about meiosis, so let's get started. Okay, so to introduce the topic of meiosis, we have to talk a little bit about reproduction in general. So there are kind of two main types of reproduction that we discuss in an intro biology classroom, and the first one is asexual reproduction. So in asexual reproduction, the offspring are going to be genetically the same as the parents. Usually it happens in unicellular organisms that can potentially reproduce by binary fission, or maybe in just meiosis without the need to make any haploid cells or gametes. And oftentimes asexual reproduction in the biological sense is very efficient in terms of energy because you don't lose any energy looking for mates, doing any behavioral displays, etc., and you pass on all of your genetic information to your offspring. So in terms, again, of genetics, the offspring and the parents are genetically identical in terms of asexual reproduction. So, some animals perform this, and also bacteria obviously go through this type of reproduction. But we're going to be talking a little bit more about sexual reproduction because that's the one that's perhaps more interesting, right? And in sexual reproduction, the offspring have the chance to be genetically different from the parents. And each parent contribute to one gamete. Those gametes will ultimately fuse together to form a zygote. And depending on how things are sorted or segregated, you can actually lead to great genetic diversity. And this genetic diversity can be very useful in contributing to rapid evolution. So, when it comes to these concepts, however, you have to help differentiate the two types of cells. So within your body, there are somatic cells, which are not specialized for reproduction. These contain a homologous pair of chromosomes with the same genes, and one homolog of each homologous pair comes from each parent. But again, they're not contributing to reproduction. They're going to be contributing to other bodily processes. Now, gametes, on the other hand, are your other type of cell, and these are specialized for reproduction. So gametes are very fascinating because they only contain one set of chromosomes, one homolog from each homologous pair. And these gametes are going to be formed through meiosis. So the topic we're discussing is meiosis, so we obviously have to discuss gametes in brief. Of course, when you talk about meiosis, you also have to talk about genetic sets, or the sets of chromosomes within the cell. So ploidy is this idea that can be used to help identify the number of sets within a cell, so number of chromosomal sets within a cell. Generally speaking, our cells are often diploid, so there are two copies of each chromosome that form homologous pairs, and this is what's often found in somatic cells. So this is a good example in humans where the vast majority of our cells are diploid, or in the 2N configuration. There are also haploid cells, which means that the cells contain only one copy of each chromosome. And haploid is usually designated as N, so it's one set. And again, this is what's generally found in our gametes. Fertilization then is when you have two haploid gametes come together, so in our case it would be an egg and a sperm cell coming together and fusing to form that diploid zygote. So again, when you think about reproduction and sexual reproduction in, for instance, mammals and humans, you're making egg cells in females and sperm cells in males, and you're fusing them together to form the zygote and ultimately reforming that diploid cell via the fusing of two haploid cells. So that should make pretty straightforward sense, I'd say, and this is something definitely to keep in mind in terms of genetics. In terms of meiosis, however, you have to break things down just a little bit more with the different steps. So the overview of meiosis is basically you're going through one round of DNA replication. So think back to your cell cycle with the G1, the S, and the G2 and M phases. So remember, in the S phase, you have DNA replication. that happens in a meiotic cell as well. But while you have one round of DNA replication, there are actually 2 rounds of cellular division. So you start off with one diploid cell that's in a 2N configuration, and you're going to end up with 4 haploid cells in an N configuration. This is very fascinating because afterwards you can have different segregation and different assortment of your specific chromosomes, and that can lead to genetic diversity among the gametes, which is obviously very, very important. So now let's go through the actual steps of meiosis. So the first step of meiosis is prophase 1. There are some important components and events within prophase 1 that you should probably get to remember or memorize / apply. Should be pretty straightforward, I'd say. But one thing that you're going to see is the formation of the centrosomes, and they're going to start migrating to the opposite ends of the cells, similar to what happens in mitosis. However, what's different for mitosis is that you have homologous chromosomes that come together and form this pairing. So this process, synapsis, is when you have those chromosomes that are homologous coming together and forming a pair called a tetrad. So this tetrad formation is this idea of 2 homologous chromosomes coming together and pairing right next to one another. The chromosomes might be different in terms of minor loci, but in terms of the overall length, they're going to be the same. Now, it gets a little bit complicated because we also have this thing called a chiasma or chiasma, and this is where crossing over really occurs. So crossing over between homologous chromosomes occurs around, usually between prophase 1 and metaphase 1, but you have this possible exchange of genetic information between those non-sister chromatids in the homologous pair. So this section of the chromosome might switch over to this section, and this section might switch over to this section. So this occurs during prophase 1 all the way up until around metaphase 1, and it occurs in the chiasma. And this is very important because it increases the variability of the offspring that you will see, potentially. Now we move on from prophase 1 to metaphase 1. And in metaphase 1, really the main key event is that the homologous pairs line up at the equatorial plate, like so. So you can see there's a little bit of chiasma action happening there, some crossing over potentially that's finishing up. And it is going to have this lining up of those homologous pairs of chromosomes on the equatorial plate. So that's metaphase 1. Fairly soon after metaphase 1, we hit anaphase 1, where we see the separation of those homologous pairs, and the chromosomes actually still have the sister chromatids stuck together. So if you think back to mitosis, the one round of division in mitosis separates the sister chromatids. But in meiosis, however, that doesn't happen. You're separating the homologous chromosome pairs in the first division, but the chromatids and the centromeres are still there. So the 2 chromatids are still attached by the centromere, and these 2 homologous chromosomes move to opposite sides, and basically it makes it so that each homologous pair of chromosomes are separated. So you have originally that tetrad, now you're just going to separate that tetrad, which is what's shown here. So you end up with sort of 2 different ends of the cell, which means you get to telophase 1. And in telophase 1, the chromosomes are separated to the poles, like so, and the nuclear envelope may reform, and cytokinesis follows to basically form that cleavage furrow to separate the different nuclei at this point into 2 separate cells. So you have basically 2 separate cells at the end of telophase 1 and basically at the end of meiosis 1. So to recap, at the end of meiosis 1, we basically turned 1 diploid cell into 2 haploid cells. So remember, this right here is a haploid cell, this is a haploid cell, because we basically broke the total amount of genetic information down by half. But what's really fascinating is that you're actually not going to be replicating anything. So at the end of meiosis 1, we create 2 haploid cells with those replicated chromosomes here, but we're not going to run another replication before we get into meiosis 2. So there's only one round of replication. So think back to that slide that we talked about earlier, where there is only going to be one round of DNA replication, but 2 rounds of division. So if you get a little bit confused by that, just think about how you basically have originally 2 sets of chromosomes, you split that down by half, and so now the remaining cells at the end of meiosis 1 are going to be haploid. So after meiosis 1, you already have 2 haploid cells. But these 2 haploid cells still have chromosomes that are basically sister chromatids, so you could break them down even further, which means you go through another round of basically cell division. And in this case, it's through prophase 2 and then all the way down into telophase 2. So you go through meiosis 2, and in meiosis 2, you're going to be separating those sister chromatids. But in meiosis 2, you start off with prophase 2. And the main key event of prophase 2 is, once again, condensation of those chromosomes and then the assembly of the spindle, which is quite similar to what we saw in prophase 1. In metaphase 2, something similar also happens where you line the chromosomes at the equatorial plate again. But this time you're not lining homologous chromosomes because those are already separated. You're just taking those individual chromosomes, lining them up on the equatorial plate and splitting them apart, which is going to be what's happening next. But for just metaphase 2, you're just going to be lining them up on the equatorial plate. Then in anaphase 2, you're going to split those chromosomes up, and so each chromosome becomes technically two chromosomes, and the sister chromatids are going to be pulled apart through this really long, complex series of interactions between proteins, and they migrate to the poles after anaphase 2, which means we hit telophase 2. So in telophase 2, once again, those chromosomes are going to decondense and the nuclear envelope is going to reform. So ultimately, we end up with 4 cells at the end of meiosis 2. So the final product of meiosis is you're going to be turning that original 1 single diploid cell into 4 haploid cells, which eventually can basically be changed into gametes. And each individual cell has its own specific genetic variation based on concepts such as the law of segregation as well as the law of independent assortment. And then obviously when you talk about things like recombination, etc., that can also lead to certain amounts of cells having recombinants and then others maybe not having recombinants. So that's basically one more step into the question, but that's more of a genetic Mendelian genetics topic that we will discuss perhaps in the future. So why is there going to be genetic diversity? Well, the recombination that we saw in prophase 1 affects genetic diversity because the formation of the chiasmata or chiasma is quite random, and their crossing over can allow for that exchange of genetic information. In addition, the independent assortment in metaphase 1 also allows for genetic variation. So how are those chromosomes going to be aligned? Are the homologous chromosomes going to be like this? Or are they going to be flipped over like this? So depending on how these are going to be lined up, you can basically have this result where this chromosome here might end up in this cell if it's lined up this way, or if it's over here, it might end up in this cell. So that can also allow for basically genetic diversity. So how the homologs line up on the equatorial plate and the combination that goes to each daughter cell is quite random, and there's also a little bit of chance in regards to that orientation, which can lead to ultimately genetic diversity. Now, I can't talk about meiosis without talking a little bit about errors that can occur in meiosis, which can happen. And one is aneuploidy, which is a lack or excess of chromosomes in basically an error event in meiosis. So this can result in various mistakes. For instance, homologous pairs fail to separate at anaphase 1, Or there could be instances, for instance, where sister chromatids fail to separate at anaphase 2. Basically, what can happen is you can have this result where one of the cells, for instance, this cell over here, that gains one extra chromosome than it normally should have. And what that can mean is that with these errors in meiosis, you can basically get extra chromosomes. So an aneuploidy is a lack or excess of chromosomes. And if you have an excess of chromosomes, for instance, on chromosome 21, you can form a trisomy 21, and that means that you have an extra chromosome on the 21st spot, on your 21st chromosome, and that can result in issues such as Down syndrome. In addition, you can have things like translocation. So a piece of a chromosome can break and actually attach to a non-homologous chromosome. So that can get very messy and can also be very lethal. But what's really fascinating perhaps about some of these errors such as translocation is that it can potentially change the overall morphology of certain organisms should that issue, that mutation, that error in meiosis be non-lethal. But oftentimes errors in meiosis can lead to various diseases that can be detrimental to the organism. This is a pretty straightforward topic. Usually it gets a little bit complicated if we start talking about, diversity and genetic recombination. But overall, I think the processes in regards to meiosis is fairly straightforward. It's very similar to mitosis, it just kind of adds on a little bit of extras. And if you have a good idea of mitosis, meiosis shouldn't be that much more complicated. So once again, I hope you all found this video useful, and as always, best of luck studying, and I'll see you potentially on the next video in the future.